I'm Vidyanath Chaudhary and I'm working as a postdoc fellow at Hospital for Special Surgery. Today I will introduce our recent work on how mechanosensing regulates the plasmacytoid dendritic cell in the skins, which is recently published in Journal of Experimental Medicines. First of all, I will talk about the rationale for this work and the graphical representation here, and you are very welcome to look at the raw data. It is well known that nucleic acid sensing is very critical for type 1 interferon production during viral infections, and there are multiple players. This is a review by Marco Colon in which it is well summarized that PDC, which is dependent on the TLR7 and TLR9 ligands, are in few number but produce very high magnitude of type 1 interferon before other immune cells come into play. So what we are trying to propose that PDC and the other immune cells are contributing to autoinflammation and autoimmunity. And the key is to pinpoint the role of the PDC in the specific organs during the timing of response. We believe that there is clear evidence suggesting that the PDC plays dominant role in cutaneous lupus and scleroderma patient, which is the focus of our research. This is a clinical trial study where they have shown that by targeting the PDC using the BDCA2 antibody, there is transient impact on the interferon response change in the blood, but there is a complete abrogation of the interferon response change in the skins. In addition to that, they also observed that there is a complete abrogation of the skin lesion in the skin of the lupus patients at 4 weeks, suggesting the dominant role of the PDC in the skins. We got interested into the scleroderma patient, which is a complex disease and characterized by fibrosis, vasculopathy, and autoimmunity. And it is well established that there is a skin thickening and severe stiffness changes in the pigmentations. We and others have demonstrated that PDC infiltrated into the fibrotic skin of the scleroderma patient by using the different marker for PDC. In addition to that, we have also demonstrated that by depleting the PDC in the skin fibrotic mice model, there is a reduction in the skin thickening and the interferon response genes in the skins. This result was further confirmed in the humanized mice model by targeting the PDC with the BDCA2 antibody, suggesting its role in the skins. So the two questions we are trying to address is that what is the impact of the skin on the PDC function, and why these PDC are chronically activated in skin inflammatory diseases once the PDC reaches the skin during the disease conditions. So this is the graphical representation of our work, where when there is an injury or the inflammatory signal, the PDC are recruited into the skin, which is localized in between epidermis and dermis, and these PDCs communicate with other immune cells and the non-immune cells, such as like macrophages and fibroblasts, that lead to the fibrosis and increase in the stiffness. The increase in the stiffness then reduces the interferon response by interferon response in the PDC. That result in the normal wound healing process by activating the NRF2. But in the disease such as scleroderma patient, the presence of the CXCL4, which can bind with the DNA, can disrupt this process and lead to the chronic inflammatory response and persistent fibrosis. And please look at our data, and if you have any questions, contact me or the senior author. Thank you.